All right, good day, class. So this lesson is on multiple alleles. Okay, so let's begin. Still part of non-Mendelian genetics. So what are multiple alleles? So the information that a gene carries is extensive. So what the gene stores is a very extensive. Okay, and even mutations can modify. It can modify this information in many ways. So what the mutation does is it alters or it can produce a different allele. So therefore, for any gene, the number of alleles within members of a population need not be restricted to two. All right, so it need not be restricted to two. When there are three or more genes, or three or more alleles rather, take note, three or more alleles of the same gene, we call this multiple alleles. So these are found in a population, and the resulting mode, the mode of inheritance, is quite unique. But never ever forget, okay, you have to realize that multiple alleles can be studied in populations. There may be several alleles for one gene. However, we call this only alternative forms of the same gene. Okay, these alternative forms of the same genes can exist. Okay, and do not forget that for a certain trait to be expressed, again, a pair is needed only. So of the several alleles that can represent a certain gene, do not forget that a gene can be expressed when in pairs. Okay? Uh, so any individual diploid organism has at most two homologous genes that may be occupied by different alleles of the same gene. So an example of multiple alleles, of course, is the feather pattern of mallard duck. So look at this presentation. You see here the mallard ducks. One allele M produces the mallard. All right. So, all right. So the M here, I just got my pen. The M stands for mallard. So that's the common thing, mallard ducks. But another allele, M, with a superscript R, gives you another phenotype which is restricted. So in this picture, that's the brown one. Okay, that's restricted. So, and then without the M, uppercase M and the uppercase M with the superscript R, you have the mallard duck, the dusky rather, which is the recessive allele. So you will see that there are three alleles for the gene which determines the duck feather pattern. And you have to check the order of dominance. So the most dominant is restricted, followed by mallard, followed by dusky. So what do you notice? If in the gene pair, so you see there's still two alleles per gene pair. So when the M superscript R is found, the phenotypes are all restricted because M superscript R rather, or the restricted, is the most dominant. Now, when there is the absence of MR and you are only left with a M or uppercase M, that's when the mallard phenotype appears. But when the M, uppercase M, is not present and you have the homozygous recessive M superscript D, okay, you now have the dusky phenotype. So for example, you cross a mallard duck with a dusky parent and you cross it with a restricted duck who had a mallard parent. So what was that again? A mallard duck with a dusky parent crossed with a restricted duck who had a mallard parent. So yung mallard, automatic uppercase M. But then this mallard duck had a dusky parent. So heterozygous. And then you have a restricted duck who had a mallard parent. So that's when you place this. You see, so you also need to know how to represent your parents or parental genotypes properly. So doing Punnett square Okay? You will have restricted, mallard, restricted, and mallard. So that's 1:1 or 50% restricted and 50% mallard. All right, so no dusky appeared. Okay, so let's try this. The ducks in the following list are crossed to produce the offspring shown. Give the genotypes of the parents and offsprings in each cross. So you have a restricted and a dusky parent, and the progenies produced are one half mallard and one half restricted. So what could be the probable genotypes of the parents? So since I already solved this in advanced class, for the restricted and the dusky to produce one half mallard and one half restricted, then you should have a gene for the mallard. Kasi di ba you have a restricted duck, so you have to have a mallard gene, and then you cross that with a dusky. Remember that a dusky is recessive, so that's very easy. This is dusky. So if you do the Punnett square, you will have anything with MR, anything with MR is restricted, and then anything with M will be mallard. So that's correct. One half restricted, or 2 out of 4, and then one half mallard, 2 out of 4. All right, so you got that. So the next problem is restricted times mallard, but then a dusky appeared. Para class kasi maging madali yung pag-solve nyo, i-analyze nyo na din yung progeny. So what we are doing actually here is a back cross. Remember, sa review nyo, when you do a back cross, what we're looking for are the genotypes of the parents. So we have here, you have a restricted and you have a mallard. Take note of the progeny. Merong mallard at merong dusky. So hindi pwedeng ang restricted mo ay pure. It must have at least a either a mallard gene or allele, and then or a dusky allele. Alright? So, para magkaroon kasi ng dusky, remember, you need 2 genes for that. So, dapat po, yung isang parent ay kailangang mag-donate ng dusky. Then, the mallard, on the other hand, this mallard parent must also donate a dusky gene for a dusky to be produced. So from there, in a back cross, alam mo na yung magiging genotype ng parent even without checking or solving using the Punnett square. But then just to show you, so this is the restricted parent and this is the mallard parent. So let's check if the ratio for the progeny is correct. Always write the dominant first. All right. So you will see here, this is a restricted, so that's another restricted, so that's 2, and then 1 mallard, and then 1 dusky. So 2:1:1, or in other words, 1/2, 1/4, 1/4. Okay, so that's the answer for letter B. So how about letter C? You have a mallard and a dusky, but then in this case, all the progenies are mallard. So, hindi dapat lalabas ang dusky. So, that's very easy. You should have a pure mallard and you cross it with a dusky. So, by simple cross, you will have 100% mallard ducks. All right, I hope you got that. Let's move on to the next slide. We have another type or another example of multiple alleles is found in the coat color in rabbits, as shown in this figure. So you have an agouti, a chinchilla, a Himalayan, and an albino. So you will observe here that there are 4 alleles. That's uppercase C, CCH, CH, and C. In other references po class, iba din yung pagka-represent nito. But in our case, we will be using the C for agouti, C superscript CH for chinchilla, C superscript H for Himalayan, and the lowercase c for albino. Okay, so the chinchilla phenotype, you will see CH. So that's chinchilla. Okay, uh, that's the one with the this one. Chinchilla is the gray one. All right. Now, the albino, as you can see, is colorless, so it's all white. And then you have the Himalayan. Uh, you will observe that the Himalayan have black fur only on the extremities. So, the snout, and then the limbs, and then the ears. Okay. And you notice from our lesson on gene interaction that this Himalayan expression, rather, of the black tip fur is also affected by environment. So, um, the complete dominance of the agouti, therefore, so this is the pigmented gene, may be affected by mutations or other mutants that can occur as an effect of um, dosage compensation on yung gene product, or we call this penetrance, and the one I mentioned before, the expressivity. Okay. Um, you will see now. Let's try. So what are the phenotypes in this case? Okay. Now, coat color. For full color, so you will see agouti full color, all uppercase C are found. For chinchilla, CCH if it's present, and wala na po si uppercase C. For Himalayan, the one way if H na lang ang present and not the CCH. And of course, albino, which is the most recessive in this uh, series, will only appear if it's homozygous, right? So let's try some examples. What types of offsprings could be produced by a full color rabbit that had a genotype of CCH? Sorry, CCH. So we have CCH. Bred with a Himalayan rabbit that was CHC. So that's very simple. Okay. So you have to be very careful with this. So anything with an uppercase C, of course, will be a full color or agouti rabbit. And anything with CCH, so you have the CCHC and the CCHC, even if this has H, still it's gonna be chinchilla. So you will see that half or 1:1, half, one half or 50% of your progeny will be agouti, and also one half or 50% will be chinchilla. All right. So moving on, more practice. Predict the phenotype for a rabbit with a CHCHC genotype. Of course, the presence of this makes it chinchilla, even if it was written in reverse. The phenotype for a rabbit that's CCH. Of course, the presence of uppercase C makes it agouti or full color. And then you have, would it be possible to obtain white rabbits if one parent is white and the other is chinchilla? What do you think? The answer is yes. So you have a white rabbit that's definitely homozygous white. And then the chinchilla should be with the phenotype CCHC. Okay? So the chinchilla rabbit must carry uh, an albino gene as well, or heterozygous siya for the white gene, okay, which is it's albino. So if you segregate, so you will have a chinchilla, and half of the progeny will be albino. So that's one half chinchilla and then one half albino. All right. So next question will be, would it be possible to obtain chinchilla rabbits if one parent is Himalayan and the other is white? This is a no-brainer. In the order of dominance, you have agouti, dominant to chinchilla, dominant to Himalayan, dominant to albino. So if you are crossing a Himalayan and an albino, where will you get the chinchilla gene? You can't get any chinchilla gene. So, which means that this is a big no. All right. And last, a chinchilla is a rabbit is mated with a Himalayan. Some offsprings are white, So what do you think are the parents' genotype? So what we're doing again is a back cross, a chinchilla rabbit. So remember that for a white to appear, then the chinchilla rabbit as well as the Himalayan must donate an albino gene. So which means that your chinchilla is a hetero and carries the albino gene, and your Himalayan as well is hetero and carries the albino gene. So by crossing your chinchilla and your Himalayan, you will have a chinchilla, another chinchilla, a Himalayan, and one-fourth albino. So that's one half chinchilla, or 2 out of 4, one-fourth Himalayan, and one-fourth probability, or 25% that you have an albino rabbit. Okay, very easy ang multiple alleles class. Okay, in some cases, there will be problems wherein it's a combination of multiple alleles and codominance, or sex-linked and codominant. So you have to be very careful if these problems appear, right? In this one, maybe we'll just try a few of this problem. Of course, this comes from your book club. In guinea pigs, one locus involved in the control of coat color may be occupied by any of the four. So multiple alleles, four. C is full color, and then CK is sepia. So you have sepia, full color, and then CD is cream, and then you have CA is albino. Again, the absence of color, with, of course, the order of dominance going there. All right? Now, let's try sepia, letter A. So you try answering this in your book as well. I think some of the items have been answered. So you look for the answers at the back or the latter part of the ebooks if you have time. So sepia and cream are guinea pigs. So this is coat color in guinea pigs, all right? Had an albino parent. So si sepia, so that's CK. Kung meron siyang albino parent, then ganito. All right? So the cream, so that's CD, also had an albino parent. So ganun po yun, class. Okay? Because the other parent donates the gene. So it means that they are both heterozygous for the albino gene. So let's place here your sepia and then your cream. So what is the probability? Always write the most dominant first para wag po kayo malito. So this is, so this one with the CK at the beginning. So that means you have sepia. 50% or 2 out of 4 or 1 half is sepia guinea pig. And then you have CDA, that's 25% or 1/4, that's cream. And then you have also 25% or 1/4 albino. All right. Okay, let's try letter C. So let's try to skip, try to answer the rest. So we have a sepia and a cream. So that means CK. And what's cream again? That's CD. All right. Where the sepia, guinea pig, had two full color parents. Oh, if the sepia guinea pig had two full color parents, it means that your sepia is pure. Kasi ang full color class, C times C, pwede lang yan magkaroon ng anak na sepia kung hetero sila for the CK. Okay? So from here, ang mapoproduce nito ay full color and sepia. So it means that if a sepia had two full color parents, then the sepia parent here is pure. And the cream had two sepia parents, okay? The same. Si cream is CD, mas dominant si sepia. So what does that mean? If your cream guinea pig had two sepia parents, Ano kaya siya? So, ang dalawang sepia, CK ay pwede rin lang magkaanak ng cream kung ganito. So, ang magiging progeny nito ay 50% sepia, um, 75% sepia, and 25% cream. Alright? So, this is genotypically 1:2:1, but this will be all sepia. So, asan dito yung cream? So, eto po siyang cream. So, 25% probability siya. So, that is just to show you the origin of a particular genotype. So, dapat alam mo siya so that you know whether a new parent, in this case, sepia and cream, is homozygous or heterozygous. So, in this case, the sepia is homozygous kasi coming from two full parents siya. The cream is also homozygous because this cream came from two sepia parents. So, no brainer, no need to solve. All the progeny will still be sepia. Kasi po, sepia is dominant to cream. Okay, so the progeny for this cross will be 100% sepia. All right, so moving on. Oh, it's over. So this class is the end of multiple alleles. So another example, so a continuation in another PowerPoint is blood typing. So remember that blood type is also an example of multiple alleles as well as codominance.